So one of the reasons why we cover Mendelian genetics and chromosomal genetics and try to look at the patterns in meiosis and so forth is so that we can really start to understand mechanisms of evolution. So now when Charles Darwin proposes natural selection as a mechanism of evolution, of change over time, descent with modification, his words, not really evolution, at least initially. The idea is, you have, so for example, with our tall and short plants, right, that some they have genetically just are taller than others in this particular example. The taller ones, let's say they're growing together, are going to receive more light from the sun. The shorter ones are going to be more so in the shade, and they're not going to receive as much light. That means that the taller ones may then have more energy that they can put into reproduction, which means they may have more offspring, and the shorter ones may not, actually. They may not have as much energy. So although they may still be able to produce offspring, they may not be able to produce as many. And then in a future generation, the alleles, the frequency, how often we see the tall allele versus the short allele may change, because we'll see fewer, say, shorter alleles and more of the tall alleles. That is then the basis of what we're trying to get at. So the scope of this particular lecture and the next one or two beyond it are all going to be to kind of look at following alleles and the frequencies of alleles in a population over time, seeing whether they change or remain the same. Now, if they remain the same, the population is in equilibrium, saying the frequency of alleles and genotypes in a population remain constant. It's part of the Hardy-Weinberg principle. And that means no evolution is taking place. The population is just stable, and everything, genotypes and phenotypes from one generation to the next, are staying the same. However, if something is acting upon those populations, which could be a whole variety of things, which we'll also get into later, like mutation and or selection and or a whole variety of other things, drift or input from other different populations, then things will change. And so that's why we're going to also kind of get this term a little bit here is population. We're now focused on population genetics, not the genetics of an individual, but of a whole population of one species. So we have a species of organism, not multiple species, we're just focused on one, and they have some defined region or border. So we're looking at those individuals belonging to a species within a specific area that are not breeding with other individuals of other populations in different areas. There's some kind of boundary or restriction that keeps them from mixing with one another. So we have a set group is the idea. This could be done in a lab setting, but we're trying to talk about it in the wild. We have some terms that we have to introduce. We talked about genotypic frequencies and phenotypic frequencies when we talked about Mendelian genetics. And now what we need to talk about are allele frequencies. Okay, so we'll look at a couple that we worked with before, or that I used as examples, and then we're going to kind of change the example slightly, sort of expanding on it and hitting on some other topics as well in genetics, and using it to see how we could actually score or measure allele frequencies in a population. So now let's say we had this example before with red and white flowers. Okay, so this is just a different example. And with the red and white flowers in our example, the red was dominant, and the white is recessive. Okay, and so we had like this. So now for the recessive one, we know their genotype because they have to be, if you remember, homozygous, or else you won't see that characteristic or trait. The dominant phenotype, however, we don't know their genotype, right? Because they could be heterozygous like this, or homozygous like that, or it could be heterozygous and they would both appear red, right? So we're going to then introduce a variation of this where we're going to have incomplete dominance and mix the two and actually be able to then produce pink flowers. And then that would shift over to be the heterozygous genotype, like that. Okay, so this is an example of what's called incomplete dominance. It's where, this one isn't recessive to the other. And so in that particular case, then we don't use this designation anymore. The capital letter and the lowercase letter, it's not used because it's not strict dominant recessive relationship. So we use something else. So in this particular version, what we do is we have color gene like this for the flower color. And then we're going to still use R for red like that. But what we're going to do then instead is use W for white like this. And so the white ones have to be white, white. The red ones have to be red, red. And then the pink ones will be the heterozygous ones. And they'll be red, white. Okay, so this will be our new designations, right, for our genotypes. And these are going to be the examples we're going to use with this incomplete dominance and flower color. And I'm going to use it for a few different examples of how to show the Hardy-Weinberg equation, how to use the Hardy-Weinberg equation, how to calculate frequencies of alleles in a population, and then how to make predictions from things that we observe in a population of the allele frequency. So to go both directions, sort of alleles to individuals in the population and back and forth. So we have to kind of get at the terms and the frequencies and how they relate. So if we had, say, two pink individuals here in a population, let's say, so we have to get both genes, right in here, so, or both alleles, sorry, both versions of the allele. So we have the version for red and the version for white. And if we bred these, we would then have some individuals show up who are red. Some individuals show up who are pink. More who show up who are pink. And some who show up who are white. And we would see our traditional 1 to 2 to 1 phenotypic ratio. What we want to do is look at, and if we looked at frequencies, right, then this would be, 25%, 50%, 25% of the population, and a frequency is another version of the percent. So this would be, say like 0.25 to 0.5 to 0.25, and together they equal 1, or that's 100% of the population. That's kind of how we're going to be relating to these terms. Now, if we just look at, though, alleles, right? And so we look at, say, the red allele. If we look at this group here, we have 1, 2, 3, 4. So we have 4. Just in this box, we multiply this out. If we had hundreds of individuals or thousands of individuals, it's still going to be the same proportions, right? So we have 4 for the red. And if we go for the white, 1, 2, 3, 4. Okay, so we have 4 for the white. So that's 1 to 1 as far as a ratio goes, but in terms of the population, that's half of the alleles in the population are red, half of the alleles in the population are white. Okay, so we have 0.5 for the allele frequency, that's half. Our red, 0.5. Are white. And so that's for 100% of the population. Okay, so let's try to do another example here. Let's say now we have actually a red individual that we're going to mate with a pink one. So we have red here and a pink here. We're going to see red, red, pink, pink. Okay, so in terms of phenotype, right, we have a 50-50 ratio, right? 1 to 1 in terms of phenotype. These are red, these are white. But if we're to look at allele frequencies, we come up here for reds, okay, so for C R. In this group, we have 1, 2, 3, 4, 5, 6. And the white's 1, 2. So that's the same as a 3-to-1 ratio, right? And that's of the total. So this would be 75%, right? Or 0.75. And this would be 25%, or 0.25. And that would equal 1. So this is now what we're getting at here. We're calling this allele frequency. So because we're looking at the individual alleles, right, for the red or the white, because there's really only two, even though we have a third phenotype here, it's still just the two alleles. Right? And that's kind of important. It's also going to be important, you'll see later on, as we look at a population and we're trying to determine allele frequency. If the recessive allele is hidden and we can't see it all the time, especially in the heterozygous individuals like this here, if you can't see that because it looks red, then you can't, it's hard to count them. But if the heterozygous individual has its own unique phenotype, then you can actually see them in the population, and then you can start to do these sorts of calculations. So it works a lot better for especially our examples and explanations if we use something like this as our system. Now, these terms here, P and Q, where do they come in? Well, if we look at the Hardy-Weinberg equation, there are really sort of two versions of the equation to look at. If the whole population is 100% of the individuals, or 100% now of the alleles, in the population, P and Q are going to represent then the two versions of the alleles. Okay, so the R and the W. So in this case, our CR is going to equal, say, P here, and the CW is going to equal Q. So the red is P, the white is Q. And so what we should have is, right, that P, maybe I'll swap that out because it seems to be dying on me, P plus Q, okay, so the frequency of one allele plus the frequency of the other allele should equal 1. It's just a simple equation. And that just means that if you know the frequency of one, you can calculate the frequency of the other. How does that relate then to the genotypes? Well, we have two, right, of each allele. So for the red individuals, right, the CR individuals, that's P, like this. So that is P squared plus the pink individuals would be the red and the white. So that's P and Q, like this. If we come back here and look at our typical proportion ratios of a 1 to 2 to 1, then there's going to be 2 PQ, like this. And then for Q, that's the white, or just the the other allele, so that's going to be Q, and then it's white, so it's squared. There are two of them. That should also equal 1. And so this is the second equation. So this equation represents the individual alleles. This one uses the individual alleles to look at the genotypes. And in this particular case, in our example, the genotypes all have a phenotype that we can identify, which is now going to make using them in our examples a little more easy to follow. Because when we talk about the pink individuals, we'll know that they have this specific genotype, or the red individuals or the white individuals, and we can use those terms to then identify them. All right, so the next thing to do is see, how do we actually use this? And so in a very simple way, if someone just tells you, for example, that the frequency of P in a population is 80% or 0.8, and you want to know, well, what's the and that's, so that would be the red, in our case, allele. So what's the frequency of the white allele? Maybe that's our question. Well, if the white allele is represented by Q, and P plus Q equals 1, what's the difference between 1 and 0.8? Well, it's 0.2, right? 0.2 plus 0.8 equals 1, so now we would know that. So this is a very simple, sort of straightforward thing that we would do. The next part of it is sort of figuring out, from a population that we would observe, and then testing this, to kind of see, how it actually works, putting the numbers into it. And then ultimately our goal, is I'm just kind of doing this right now to give you terms, the equations, their meaning, and an example of how they just kind of come together. Then what we want to do, or not in this lecture, it'll be the next one, is then look at how other factors, then that influence the population, that cause selection, will cause changes in allele frequencies from generation to generation so that they don't stay the same. But first we have to know what they are to be able to know whether they are staying the same or whether they're changing. If we don't even know what they are to start with, then we can't monitor their change over time. So we have to do this part first where you can actually figure out what are the frequencies. And once you know the frequencies, then we can go ahead and compare them, between populations over time and determine whether there is change or no change, and then determine whether those populations are evolving or not. And so that brings us to this other term here, microevolution. So that's going to be the change in allele frequency over time. So if Hardy-Weinberg principle is running and working and it's in equilibrium and there is no change over time, that means no evolution is occurring. But if there is change, if there's any change in the frequency of alleles in a particular population from one time period to another, then that means microevolution has occurred. This is one of our other things in science that people sort of have a problem with over time, is some of the use of terminology versus common everyday use and the way we use it in science. So a theory many people think is, it's their idea. Oh, I have a theory about this or that. But in science, we use the term in a different way. Theories are accepted as an explanation. They are supported by evidence. Your idea isn't. Your idea is just an idea, but you'll use the word sometimes theory to represent the idea. And then people then think scientific theories are just unsupported ideas, and that's not true. So we use the term in a different sort of way. Same thing, when people hear the term evolution, they often think major macroscopic change from one type of, from a fish, to an amphibian, or massive, and that is a type of evolution. But evolution is defined on the smallest scale. What leads to all those massive changes that we can then see because they're so dramatic is the genetic changes. You have to have the genetic changes first before you'll see those other major phenotypic changes and big changes in organisms. So that's why we're focusing on this. We have to look at the allele frequencies, the genetic changes that occur, and that's what supports our bigger changes. All right, so let's just do this quick example, and then we'll actually move on to defining some other things that then affect and influence the changes that will occur, which we'll do in the next lecture. So let's say, for example, we have a population of flowers. Okay, so we're looking at a population of these flowers, and there's 1,000 flowers. And in that population, we do a quick survey and we count 350 red flowers. So applying it to what we know so far, and that's all we counted. We didn't maybe we don't count the others at first. We're just going to try to do some predictions. So what do we know? So that means that red is represented by the R allele. Okay, so our CR is red, and that's also represented by P squared. All right, and so 350 out of 1,000 is 0.35, or 35%. So now we know that P square is 0.35. What we really want to do now to get at these other numbers, though, is actually we have to go here first. So what we really need to know is p. What's p? So we have to do the square root. So we go 0.35 square root is 0.59. And then we know here from this equation, p plus q equals 1. So that means we can use this equation to come back to here to solve this. What is q? So what is w? So q is going to be just CW, just the white allele. It's going to be able the difference between 0.59 and 1, which is 0.41. 0.41 plus 0.59 equals 1. So we have that. Now, if we wanted to say, for example, how many pink individuals should be in this population? I mean, you can go there and count them too, and you probably should, to then see if your numbers actually work out and if all this works out. But what we have is, so p squared, we already know, right, is going to be 0.35. Q squared, if we work it out, 0.41 times 0.41, it's going to roughly be 0.16. So the pink individuals, right, are represented by 2pq. Right, so what's 2pq? Well, it's 0.59 times 0.41, right, times 2, which is going to be 0.48. And if we work that out, this works out to 0.99, but we're kind of doing some rounding there. Okay, so it should work out to basically to 1. Okay, so that means pink individuals, I was doing this, pink individuals would be 48% of that population, or say 480 out of the 1,000 would be pink. And this is just one way of using this to sort of go back and forth between the phenotypes, the genotypes, the alleles, the frequencies of alleles, the frequencies of genotypes. So all these things could be interchanged, but where it really starts at is the definitions of the terms, and knowing what does P represent, what does Q represent. And that's really the main point of this whole first lecture, is for you to identify the terms, to identify them within the equations, to see them as part of an example of something you could relate to, like flower color. Okay, so in our example, we had flower color being red, pink, or white, but we only have 2 alleles, right? The alleles for red are white, and we are using incomplete dominance here so that our heterozygous individuals are blended and have this pink phenotype. And so we have the homozygous individuals here for red, homozygous individuals here for white, and the heterozygous individuals here for pink, and that's what we're representing here in this equation. So red, pink, white. And again, making an observation of the population, we can actually make a count and actually count the numbers. We can also then, which even more importantly, we'll get with this later on, counting the pink individuals, in the population. That way we know where the heterozygous ones are, so we can get those numbers and be more confident in them. So that's this first part, definition of terms and definition of some of these concepts. The next thing we're going to do is look at other things. Well, first off, we'll look at what are the requirements, the things that must be true for this to remain constant, and then what sorts of things affect those so that they are not true, all right, and then tend to change the population over time, and we'll look at the definitions of terms and some concepts, but knowing this first will make all those other concepts make a lot more sense.